It wasn't easy to narrow our speakers down to four. All of our ARC scholars actually have amazingly fascinating stories to tell. But these four are representative of a wide variety of research and disciplines at both WSU and at the UW. They will each tell you briefly about their work and how the ARC Scholar Award has made a difference in their lives. So our first scholar is Erin Mahoney here, who is in molecular plant sciences from Washington State University. Next to Erin, we have Irene Zhang, who's a computer science and engineering student at the University of Washington. Next to Irene is Gabrielle Pastenkos, who's in veterinary science at Washington State University. And finally, Michael Dysand, who's pursuing his MD and PhD in genome sciences at the University of Washington. Please give them all a warm welcome. So Erin, the stage is yours.
Good morning. Good morning. Thank you. My name is Erin Mahoney. I'm a third-year ARC scholar and PhD student. I'm in the molecular plant sciences at Washington State University, and I work in Dr. Scott Holbert's lab. My interest in research began during my undergrad at Wichita State University in Kansas. I worked in a lab that focused on how plants interact with their environments. This research got me very interested in plant genetics and how plants really kind of change or evolve or adapt to their environments. Knowing I wanted to continue in research, I moved to Bellingham, Washington, where I received a master's degree in molecular plant sciences from Western Washington University. Nearing graduation, I knew I wanted to continue in sciences, but wanted to focus more on crop biology and breeding. And so I reviewed all my options, and I came to Washington State University to work in wheat genetics and plant pathology. Our lab really focuses on a few of the many challenges we're facing in order to feed a growing world population. As a major exporter of wheat, the Pacific Northwest exports nearly 90% of its grains to growing markets in Asia and the Middle East. This positions wheat farmers in the Northwest with an excellent opportunity to meet this challenge. But really, for long-term production, they'll need to grow this crop in a sustainable and economic manner. To be more sustainable, many farmers in the Northwest have transitioned from conventional tillage to no-till or minimal tillage systems. Essentially, the idea of minimal tillage is that each year after the farmer harvests their wheat, instead of plowing the plant debris into the field, they leave large amounts on the field. Then in the following year, they plant directly into the soil below this wheat stubble. This type of agricultural practice has been really beneficial for increasing soil moisture, improving organic material, but also reducing soil erosion and fuel costs associated with conventional tillage. However, because there's an increase in soil moisture and organic material in the soil, this transition to a no-till system has really increased the number of soil-borne pathogens that use this material to grow and propagate. Farmers who then plant in these fields have been faced with an increase in soil-borne diseases and ultimately become more reliant upon chemical pesticides. This reliance is neither economic nor sustainable for long-term production. So in order to really address these issues and reduce the risks to the growers, my research is focusing on two different methods to increase disease resistance in commercial varieties of wheat. Our first approach is really trying to breed durable resistance from wild wheat relatives into commercial varieties of wheat using wild wheat relatives. We actually, I had got a question earlier about what I mean by that, and it's actually old grasses that we breed into current commercial varieties. From our research, we've really identified a few lines that seem very promising, and we're now in the process of identifying the mechanisms that provide this resistance. Once we identify this resistance, we'll provide this research in the lines to the wheat breeders at WSU so they can get these into their commercial programs. The second goal of my research is really understand the role of soil microbes that surround the roots of different wheat varieties. Using DNA sequencing and microbial databases, I'm characterizing this biodiversity. This will allow me to identify microbes that could be used for pathogen competition, disease suppression, or perhaps new services to the host. Really, our research is, at the end of the day, we hope it provides new ways for the farmers to adopt more sustainable agriculture, but reduces the economic losses to soil-borne diseases. Being an ARC scholar has been an amazing opportunity for myself and as a graduate student. I personally feel that I've greatly benefited from this fellowship. The support the donors and the ARCS Foundation have provided me these last 3 years has just been really fundamentally a major rewarding factor in my life. I'd like to take this time to thank the ARCS Foundation, my donors, Lynn and Michael Thompson, my advisor, Scott Holbert, and Washington State University. Thank you for your time.
Aaron, that's, the positive economic impact of your work is remarkable. If there was that one success that you'd like to see as you go through this, what would you like to see happen for our community and for our region?
Yeah, that's a great question. I think we are, in the Northwest, we're really positioned to be meeting this challenge to feed the world, with grains, from cereal, from wheat. And so, if we could just do it more sustainably and also economically, I think it would really improve our economy in the Northwest and also really benefit the world through consumption of grains. Thank you.
Great. Thanks very much, Aaron. So from wheat to computer science, Irene, the stage is yours. It'll go on in a second.
Okay, hello. My name is Irene Zhang. I'm a third-year PhD student at the University of Washington in the Computer Science and Engineering Department, working with Professor Hank Levy. Before I started my PhD, I received bachelor's and master's degrees in computer science from MIT. I also worked for 3 years at VMware, building cloud software for everyone from Lego to the White House. I was born in Beijing, China, but I spent most of my time growing up in Columbus, Indiana. So my mom is a computer scientist, so growing up, I knew that was the last thing I wanted to do. Like many childhood ambitions, that one obviously didn't work out. From the first time my mom introduced me to programming, I saw the power of software and the potential to have an impact on people's lives. I mean, more than anything else today, computers have changed the way that we live and the way that we interact with each other. So my research is in operating systems and distributed systems. Basically, I build software that makes life easier for the people who build the applications that you use every day. We're at a real turning point right now. The applications that people used to use just ran on a single computer that was sitting in front of you. But today, applications run on thousands of servers in the cloud, and they span millions of mobile devices in the hands of people around the world. This software is really hard to build. It's really hard to make sure that your data is safely stored and not lost. It's really hard to make sure that your bank account information is not leaked to the wrong people. And it's also really hard to make sure that Justin Bieber's latest tweet makes it to all of his 50 million followers. So that's where my research comes in. Every application programmer that's building an application today, they're solving the same problems that Facebook, Google, and Amazon have already tackled. So I'm building tools that hopefully will enable programmers to take advantage of that knowledge. I think there's a real potential for my research to have an impact on the people building the next generation of Facebooks, Amazons, and Googles. Right now, there's also the real potential for ARCS to have an impact on computer science at the University of Washington. So today, unlike 20 years ago, our department is really only competing with the top schools in the country for students, like Stanford, MIT, Berkeley. And choosing between those schools is a really tough decision. I know because I had to make it. And I can tell you that when it comes down to picking between the top schools in the nation, things like ARCS really make a huge difference to the students. Thank you.
Thanks, Irene. So this is clearly one of the hottest fields in the world right now. You must be getting calls like crazy from recruiters. What are you going to do? Are you going to stay in the academy, or are you going to go to the private sector? You've worked in both.
Yeah, so I'm really very seriously considering teaching, because I think throughout all the stuff I've done, the thing that I've found the most rewarding is really mentoring the younger students.
That's great. Thank you very much. Well, Gabrielle, how about you tell us a bit about your work?
My name is Gabrielle Pastenkos, and I'm a veterinarian in the Combined Anatomic Pathology Residency and PhD program at Washington State University. I work in the lab of Anthony Nicola, who's a researcher known for his work on human herpes viruses. My research focuses on bovine herpes virus 1, which is a disease that causes both respiratory infection and reproductive losses in cattle. This virus also shares many clinical and molecular features with human herpes viruses. Before coming to Washington State University, I studied animal science at Rutgers University and veterinary medicine at The Ohio State University. I wanted to become a veterinarian in order to conduct infectious disease research, diagnostics, and surveillance. And my interests are primarily in animal diseases that have a counterpart in human medicine, as well as diseases that are directly transmitted between animals and humans. Bovine herpes virus 1 is a ubiquitous pathogen worldwide. In some countries, up to 75% of cattle are infected with this virus. The prevalent strain in the United States causes respiratory disease in early infection. While most cattle do recover from the initial disease symptoms, the virus enters the peripheral nervous system and it hides in large clusters of nerve cells. So just like in human herpes virus, infection is lifelong. Neither natural immunity nor any pharmaceutical interventions can effectively clear virus from an infected animal. Again, similar to in human herpes infections, stress can induce reactivation of the virus. So just like when a person who's infected with human herpes simplex can get a cold sore after a period of stress, like starting a new job or having a stressful exam at school, cattle can become sick again. So in the beef cattle industry, a major source of stress is the transition between different stages of production, specifically when calves are shipped to the final finishing stage and are introduced to new environments and mixed with cattle from all across the country. So when bovine herpes virus 1 is reactivated from latency, the animals can develop disease that's very similar to the disease seen in initial infection, but they can also undergo immune suppression. So the virus can actually inhibit their immune system's ability to fight off pathogens. So when immunocompromised animals are exposed to calves from all across the country, they're introduced to many novel viral and bacterial pathogens. This sets them up to develop a complicated infection that we call shipping fever, or the bovine respiratory disease complex. Shipping fever is a potentially fatal polymicrobial pneumonia. It costs the US beef cattle industry between $500 million and $1 billion a year. So obviously, controlling this disease is of major concern from an animal welfare standpoint, as well as to the US beef cattle producers. So while we do know the cell that is initially targeted by bovine herpes virus 1, we don't actually know how the virus gets into this cell. My research is focused on a protein that's found on the surface of bovine herpes virus 1, known as glycoprotein B. I suspect that this glycoprotein plays an important role in mediating the initial infection of the cow. So I am lucky to work in a lab where both my mentor and fellow graduate students work on human herpes virus. One of my fellow grad students is also an ARC scholar and here today. Sorry.
It's okay, you're doing great. You're doing great. And your work is amazing.
Thank you very much. So working with people who are working on the human counterpart to this virus allows me a unique opportunity for collaboration. It also means that anything that I find out about bovine herpes virus 1 will both help cattle in the United States, it'll help beef cattle producers, it'll also help us better understand an important human pathogen. In addition to my research, I train in anatomic pathology. I'm a veterinarian, so I work for the Washington Animal Disease Diagnostic Lab in the College of Veterinary Medicine. So I analyze surgical biopsies and I perform autopsies on patients that come from the College of Veterinary Medicine, private clinics as far as Alaska and Arizona, local livestock producers, and fish and wildlife agencies. I have actually diagnosed bovine herpes virus in cases from cattle in Washington State, so my work as a pathologist reinforces the importance of what I do in the lab. And I would like to thank the ARCS Foundation and also my donors for their contribution to science at Washington State University. I truly appreciate your support, and it has definitely made my time here a lot easier, so thank you.
Thanks, Gabrielle. I just have a slightly personal question. A lot of kids, when they're in grade school or high school, say they want to be vets and they're thinking about working with dogs or cuddly animals, and you're working with cattle. This is very significant research, but did you ever imagine when you were thinking about going into this field that this would be your focus?
So I'm not a typical veterinarian. I think I never wanted to work with, I love all animals, but I always wanted to work on the disease portion of veterinary medicine. So I always wanted to work on infectious diseases, and I enjoy the livestock work a lot.
That's great, thanks. Michael.
Good afternoon, everybody. I'm Michael Dysand. I'm a fourth-year MD PhD student in Evan Eichler's laboratory in the Department of Genome Sciences at the University of Washington. I'm from the Seattle area originally and pursued studies in mathematics and chemistry at Carleton College before reading for a master's in computational biology at the University of Cambridge in the UK on a Gates Cambridge scholarship. I've always been really interested in applying analytical tools towards solving problems in healthcare and medicine. And when I was deciding where to pursue further graduate studies, whether to remain in the UK or whether to come back to the US, Seattle was particularly attractive, not only for its strengths in healthcare and medicine and genetics and genomics, but also due to other resources such as the ARCS Foundation, whom all of you represent, as well as a general climate of innovation that I found here in Seattle. And here I've really been able to pursue my passion to use my talents and use the skills and resources here to find the causes of a disorder that affects many people worldwide, autism. Why? Why is a question that thousands of parents across the US, as well as across the world, ask each day. Why does my child have autism? Until recently, we didn't know the causes of autism. But what we did know is that autism is largely genetic, and that autism oftentimes isn't inherited in families. It often occurs sporadically. So if we have a disorder that is largely genetic but isn't inherited, we can guess or make a hypothesis that children with autism must have mutations, new mutations in their genomes that are not present in the genomes of their parents. So we took this paradigm and used the most recent sequencing technologies, whole genome sequencing, to sequence the genomes of many families with autism, the parents as well as the children. And I worked on developing analytical tools to analyze these data and discover these new mutations. I just want to put things a little bit in perspective. The Human Genome Project, which sequenced one human genome 10 years ago at a cost of millions and millions of dollars, took 10 years to complete, whereas now we can sequence a human genome, any one of your genomes, for example, in 2 weeks' time at a cost of about $5,000. So we've really leveraged these technologies, and what we've discovered in these children with autism are many mutations in different genes that all lead to the autism phenotype. So this is really developing an idea that autism is actually many different disorders. It's a collection of autisms. We've also found certain genes in which families have the same mutation. So there are multiple children with mutations in the same gene. And when we look at these children and we look at their clinical presentation, the clinical presentation is almost identical. So what this allows us to do, this approach, is identify subtypes of autism, and it's allowed us to understand better the developmental trajectories of these children, as well as develop individualized treatments and therapies for these subtypes of autism. It's been really awesome in the course of my PhD to discover causes of a certain disorder and really contribute to the knowledge of science in general, as well as give hope to these families and use the newest technologies to do so. The ARCS Foundation has been a tremendous support for me during the course of my PhD and continues to be, not only financially, which is tremendous, all of your donations really help out tremendously, but also emotionally. Those late days in the laboratory, sometimes you just want to go home, but you need to finish an experiment, and it's fantastic to know that we have all these cheerleaders out here that are really rooting for us. Thanks very much.
So Michael, you said you grew up in Seattle. You went to school in Cambridge in England. Where do you hope to end up when this is all over?
Well, I certainly would like to enter academia and continue to contribute to research. But Seattle has great resources and the talent and the ability to do the research that I'm interested in. So this is certainly a place that I would want to stay if possible.
One of the things that struck me most about becoming an ARC scholar and what the intentions are of the program is actually to keep a lot of talent here in Washington State while you're doing your research, and also a chance to stay here afterwards. I just want to put to all of you, and anyone you can answer, how important is that sort of sense of belonging to this region? Many of you probably all have the opportunity to go somewhere else when you're done. Would you like to stay connected to this region? Anybody want to answer that? How important is that?
I like it here.
I'd imagine your work's all around the state, right? So it's good. How about you, Ariane? You could be anywhere.
Yeah, I have to say, I mean, coming from the Midwest, I don't miss it. I really enjoy the Pacific. No offense to the Midwest, but I made sure I could do my master's at Western so that I didn't have to go back to the Midwest. So yeah. I would love to stay in Northwest, yeah.
And Irene, I imagine the pull of Silicon Valley is just huge for your work. How do you resist? Or are you going to resist?
You know, I really like Seattle, so I'm from the Midwest as well, so I second that feeling. But I don't know, I feel like the Northwest has a nice warmness to it, that it's not as big as you know, Boston, where I came from, or Silicon Valley, which can be very impersonal. But the community here is just great, that the department is large enough that I can have lots of collaborators, and there's a lot of industrial support, but yet the community feels close-knit.
Well, I can say this for the Pacific Northwest. Thank goodness we're the Midwest. So I'm utterly inspired by the passion, hard work, and determination of these remarkable students. Let's all thank them. You guys have been great for sharing their stories with us today. I'd like to now introduce you to Dr. Lee Huntsman. He's President Emeritus of my own University of Washington and Professor of Bioengineering. Dr. Huntsman began his career in bioengineering at the UW in 1968. He's a distinguished academic leader. He served as Director of Bioengineering, Associate Dean of Scientific Affairs at the UW School of Medicine, UW Provost and Vice President for Academic Affairs, and UW President, of which now he's Emeritus. His laboratory research specifically involved the application of engineering to biology and medicine, very much ahead of his time, with regards to the measurement and regulation of the cardiovascular system. It's a level of achievement I hope all of our scholars can aspire to. Please welcome Dr. Lee Huntsman.
Thanks, Hanson. Good afternoon, everyone. It's a joy to be with you. Boy, those students, brilliant, qualified, dedicated, energetic, and articulate. How do you do that? I don't know. You know, when you're at their age, it's a real honor to be selected as an ARCS scholar. When you're at my age, it's an honor to be emeritus.